Hi, everyone. Welcome to today's Research America Alliance discussion. I'm Jenny LeRae, and as always, I'd like to thank you for joining on Zoom today and for your partnership in the Research America Alliance. If your organization is not a member yet, I hope you'll reach out to my colleague, Kristen Furlong, whose email is in the chat for information about the Alliance. Today, we are joined by leaders of the groundbreaking African Ancestry Neuroscience Research Initiative. We're joined by Dr. Alvin Hathaway, Dr. Daniel Weinberger, Dr. Kefwi Jiraza, who is also a member of the Research America Board, and Dr. Gwenaëlle Thomas. Their bios are extensive, and you can find them in the chat. We are so excited and honored to share their work with you, because it shows in very concrete terms why we need to ensure that research reflects all of us, and just as importantly, how we can make that happen through community involvement. So as always, please type your questions into the Q&A box or the chat box, and we'll get to them, as many as we can, because I think there'll be many today during our Q&A portion. So thanks again for joining us, and I'm going to turn it over to Dr. Thomas to get us started with the presentation. Thank you so much.
Thank you so much for that warm introduction. We're all so excited to be here today. And I'm going to start off with the introduction, and then Dr. Weinberger will finish the rest of the presentation. So ANRI, or the African Ancestry Neuroscience Research Initiative, is the nation's first neuroscience research initiative for the African ancestry community. Our goal is to reduce health disparities in brain research in order to ensure that all people have equitable access to new treatments. So starting out, what is the field of genomics? Genomics, or studying the human genome, this is the field that is studying our human genetics and all the personal information that makes us who we are. And as of late, the field of medicine has sort of used a one-size-fits-all for treatment, but genomics is striving towards personalized medicine. So looking at a patient's individual symptoms, their individual pathology, and trying to come up with solutions that work best for each individual. But personalized medicine is based on a reference genome. So in 2001, the Human Genome Project was published by the NIH, and this was what we call the first draft of the human genome. So the 3 billion letters of DNA that make up all of humans and make up each individual, those parts can be examined to learn more about different diseases and also the different variation among the human population. It's important to note that each human, we're 99.9% identical to each other, so that 0.1%, that is where all the variation lies in terms of disease and disease susceptibility. Minority populations are significantly underrepresented in genomic research. And so a recent analysis showed that about 10% of the African genome, so over 300 million base pairs, are actually what's omitted from universal genetic research. So you can think of it even on a broader scale when we're seeing these direct-to-consumer tests like Ancestry or 23andMe. This is omitting 10% of the African genome. And just in scale of the world, about 16% of the world is of European descent. But when we're looking at these large-scale genomic data sets that are used across research, across commercial values, this is representing 81% of European descent people. And so this is largely skewed towards European ancestry, and this also directly fits into how we see health disparities or lack of treatments for minority populations. This was a recent study that was looking at genome-wide association studies, or GWAS, for various psychiatric illnesses like schizophrenia, autism spectrum disorders, Alzheimer's disease, Parkinson's disease, and major depressive disorder, excuse me. And we saw that there were almost 5 million research subjects, but shockingly, there were 0 individuals of African ancestry. This is part of a large reason why ANRI was created, so that we can understand and increase not only the subject inclusion, but the subject diversity, so that we can start to understand how these different psychiatric illnesses are affecting people of African ancestry. And there are different susceptibilities to various brain disorders. So for example, with Alzheimer's disease, African Americans are twice as likely as European Americans to have Alzheimer's disease, yet African Americans show a different association with some of the known Alzheimer's genes. So for example, ApoE is one of the genes that's been highly studied for Alzheimer's disease, yet African Americans have a different genetic relationship with this ApoE gene. Another disease that we've studied with our ANRI project is stroke. So African Americans are 50% more likely than European Americans to experience ischemic stroke. And African American men are 70% more likely to die of an ischemic stroke. And lastly, there are some diseases that have susceptibility in the other way. So for example, Parkinson's disease is less common in African Americans than European Americans. So when we're thinking of this human reference genome, we like to make an analogy just thinking about how we have coordinates just in the world, just knowing where to travel. To find anything on Earth and in the genome, you need the right coordinates. And that's why it's so important to have this inclusive and diverse human genome, so that we can find the right coordinates for these different brain diseases, for these different neurological illnesses. We want to increase our subject pool, our research intentions, and make sure that we're being inclusive of everyone in the world, so that we can have treatments that work for everybody equally. So the African Ancestry Neuroscience Research Initiative is a public-private partnership. Our founding partners are Dr. Reverend Alvin C. Hathaway Sr., and our founding partner and current sponsor are the Lieber Institute for Brain Development, Maltz Research Labs, and the largest historically Black university in Maryland, Morgan State University. And we've also had the pleasure and honor of being supported by many different people and institutions, such as the Able Foundation, Brown Capital Management, the State of Maryland, Lieber Institute for Brain Development, and the Chan Zuckerberg Initiative. And thank you so much to our Scientific Advisory Committee, whom Dr. Jarasa is also a member of. So in ANRI, we have 4 core pillars. Our first pillar is for community engagement. Our second pillar is training and development of a diverse workforce with a focus on individuals of African ancestry. Our third pillar is public engagement in scientific research through ongoing communications and advocacy. And our fourth pillar is cutting-edge, high-priority scientific research relevant to the biological underpinnings of health disparities. And so wrapping this up before I switch over to Dr. Weinberger, we really wanted to emphasize that ancestry is the story of human history. So we know that human beings are genetically diverse, but that diversity should not be something that we run away from, but rather something that we're embracing in biomedical research. And we know that individuals of African ancestry are the most genetically diverse population in the world, and also genetic variation influences disease risk and resilience. Also, as we're emerging into more of conversations about epigenetics, it's important to also acknowledge how genetic variation is also influencing environmental contributions to this disease risk and resilience. So putting this all together, understanding how African ancestry affects the expression of genes in the brain offers a unique opportunity for personalizing medicine for brain disorders. So switching over to Dr. Weinberger.
Thank you very much, Guaniel, and thank you to the Research America people for putting this together. I think it's extremely timely, important. I'm very thrilled to be one of the speakers today. So, as Guaniel mentioned, the ANRI is focused on understanding how genes and the environment influence our brains, the development of our brains, the function of our brains, and how our brains develop illnesses or resist the development of illnesses, recognizing that genetics, our ancestors that have contributed little tiny pieces of their genomes across many generations to what each of us has as our personal genomes, these influence how our brains develop and function. So if you want to study how genes and ancestry influence the biology of our brains, there's only one way to do that. You can't take your blood. You can't measure something in your urine. You have to study the brain. If you're a kidney specialist and you want to study kidney disease, you take biopsies of the kidney, you take biopsies of the liver if you're studying liver disease, but you can't biopsy the human brain. So the only way you can study human brains is you receive donations, like any organ donation after death. And so the Lieber Institute for Brain Development, where I work in Baltimore, has put a great emphasis on getting donations of human brains for the study of brain illness. And actually, we have now over specimens from over 5,000 people who have donated brains to the Lieber Institute for Brain Development. These are from multiple centers around the country. And in the process of doing this, we've received donations from many African American families. Brains are the only way to study the molecular biology of brain illness. Molecular biology means how genes create the component parts of the cells in our brains. If I can have the next slide, please. In this initial report, we've studied 600 brain donors of individuals of African ancestry, but we have at the Lieber Institute for Brain Development now samples from over 1,200 African American families. All these donations come from the next of kin because they are of a recently deceased person. Most of our brains come from medical examiners' offices around the country, and that involves deaths that were unexpected. People that are examined at a coroner's office are examined because the death was not expected. These are not people dying in hospice or in hospital. As a result, we get brains with many different individual backgrounds. We have to ask for a donation within 24 hours of the death of the individual. It's a difficult, sensitive practice because in general, people are very, very bereft about this unexpected loss of a beloved family member. We have a team that's worked on this for many years. And the message is, this is a great tragedy, but maybe something positive can come from it, which is to do research to lessen the burden on people who are still living. About 60%, over 60% of African American families agreed to donate. And this is, I think, quite remarkable considering the history of how much mistrust there's been in the African American community with respect to scientific research. There are brains of many different disorders, but perhaps most critically unique in terms of this brain repository, because they are from medical examiner's office. We have young brains, and we have many healthy brains, people that had no brain illness at all, died in a car accident, died in some other unexpected event. So we have a large collection of normal brains. If I can have the next slide. And this led us to do the first large-scale study of how genes and the environment influence what genes are turned on and off in your brain that account for people's susceptibility to a variety of illnesses. And what we've done is we've looked at gene expression, and gene expression is measured by measuring the RNA which is the ribonucleic acid, which is the messenger that comes from the DNA to make the proteins in cells. So this is the machinery of how cells work. Genes are turned on and off across life in different cells for different reasons to build the biological blueprint by which cells in your brain work. And we looked at 4 different brain regions, which are described here. And we looked at how different variations in DNA sequence, which Gwenaëlle alluded to, we all differ in certain small pieces of our DNA, which is how come we're all different. And we've looked at what we call the epigenome, which is the machinery that regulates how genes are turned on and off. And that machinery is very much influenced by the environment. So we tried to look at how environmental contributions affect turning on and off genes in your brain, and how environmental contributions affect the turning on of gene in your brain. We looked only at what we call neurotypical individuals. This is very important. These are people that are not ill. These are people with no brain disease, because the question we wanted to ask was, how does ancestry influence our likelihood, our susceptibility to get an illness in our lifetime, even before we've gotten it? And the beauty of, from a scientific perspective, of exploring what we call neurotypical brains is that there's no confounders of illness that will systematically bias the results. The other critical aspect of this work was to avoid the minefield of comparing black and white brains. We were very concerned that if we made that kind of comparison, we would have no way of interpreting what the differences represented. Did they represent systematic environmental differences that vary across these groups? Or could we attribute them to ancestry? So the decision was made really because we had this very brilliant geneticist at the Lieber Institute who suggested that we should study principally the black brains, because most African Americans are of mixed ancestry. Since the slave trade, there's been a lot of intermixing of European and African ancestries in people who are now called African Americans. And if I can have the next slide, this is just to show you the variation. Next slide. Oh, I'm sorry. Before we get to that, the way we study gene expression is by measuring RNA, and it's called RNA sequencing. Measuring RNA allows us to look at many features that are what we call transcribed in cells in the brain. Transcribed means the DNA gives out a code to create a messenger RNA, which leaves the nucleus of the cell, goes into what's called the cytoplasm of the cell, where all the metabolic machinery is, and builds proteins. That's how the nucleus, where the DNA is, communicates with the cellular machinery of the cell to change the activity, the function, the biology of cells. And we can use RNA sequencing to measure multiple components of this transcriptional process that we call gene level analysis, exon analysis, transcript analysis, and junction analysis. And this was all part of what we did in the study. This allows us to compare how genes are turned on and off across different groups, and how genes are turned on and off based on variations in the genome. So as I mentioned, if I can have the next slide, we decided to focus only on what we call the black brains. These are people that self-identify as African Americans, because we know that they vary in ancestry. And this slide just illustrates across 292 individuals that in African Americans, about the average is somewhere between 20 and 25% of African Americans' genomes are of European ancestry. And the variation among people who self-describe as African American is from 0%, meaning both their parents are recently from Africa, to up to 60%, whereas the majority of their ancestors actually were of European ancestry. The reason this was a critical step was that because this varies across every individual, some people are 10% European, some people are 30%, 25%. This is a continuous measure. And we could use that continuous measure to predict in each person's brain how much that continuous measure, the proportion of African ancestry, determines which genes are turned on and off. And the beauty of doing that is, in each individual's brain, whether they're 30% of European ancestry or 10% of European ancestry, each brain has only one environment. So the relative proportion of European versus African ancestry is not affected by that environment differentially. So if I can have the next slide, this is a summary slide. It's very complicated and it's not easy to follow, but this circular diagram, each color is a different brain of the 4 brain regions we looked at. And what you see is across the whole genome. So this is representing all the 23 pairs of chromosomes. Across all the genome, there are many, many genes that are turned on based on the proportion of African ancestry. There are many genes that are turned on based on the proportion of European ancestry. And there's about 2,500 unique genes that are differentially turned on. That's about 10% of all the genes expressed in the human brain, a little more than that, based on ancestry. The other question we asked, well, what exactly are these genes? What are they doing in the brain? And if you look at this diagram on the right, those little red diamonds represent the biology of those genes that are turned on based on the proportion of African ancestry. And what's really fascinating about this is that these are enriched for immune function. These are about the cells and the processes in the brain that deal with viruses, that deal with other factors that could affect the immune function, the infection of the brain. What's also fascinating is that none of these genes that were based on ancestry were enriched in what we call neurons. Neurons are the communication centers of the brain. Neurons are the cells that fire these electrical discharges and that talk to each other and transfer information around. So ancestry didn't really mess with the basic machinery of how a brain worked. It messed with the machinery that defends the brain against insults. Next slide. We also looked at this question called epigenetics. So epigenetics is a molecular machinery that coats the DNA molecule and opens and closes so that the DNA can be read, so that the machinery of this cell can decide to turn a gene on and off. And this is very much dependent on the environment. And the next slide shows the same basic diagram, the circle diagram, different regions of the brain. Blue is where genes are turned on because of the percentage of European ancestry, and red is where they're turned on based on the proportion of African ancestry. And again, the genes that are regulated by environmental variation are about immune response. They're not about the neurons. So, next slide. We then asked what I think is the real take-home message here, which is, what do these genes account for in terms of whether somebody has a brain that will be healthy throughout its lifespan or whether we'll have a brain that will have other illnesses? And we showed basically that there were a number of immune problems that affect human brains that are increased in probability, up to 2 to 5-fold more likely based on these genes. But there was no preference for whether they were increased based on European ancestry or increased based on African ancestry. So there was no ancestry bias here. But that was not true for stroke, Parkinson's disease, and Alzheimer's disease. And what we showed was that about 25% or 30% of the genetic likelihood that somebody would develop Alzheimer's disease, which is thought to be twice as common among the African American community, is explained by these genes that depend on ancestry for them being turned on. About 26% of the genetic likelihood of a stroke, more common in African Americans, could be explained by these genes. At the same time, we were surprised to discover that Parkinson's disease, which we know is less frequent in the African American community, actually was predicted to be less frequent because of African ancestry in this population. The other striking finding, if you remember, I said earlier, we didn't find that any of these genes were particularly relevant to neurons, which are the cells that transfer information back and forth within your brain so that you can think, plan, imagine, control your behavior. and have been associated with all kinds of behavioral and psychiatric illnesses. Genes regulated by ancestry had nothing to do with the genetic likelihood of any of these behavioral disorders, which I think is an important context of how much misinformation there have been over decades about the prevalence of certain behavioral and psychiatric illnesses in the African American community. These data suggest this is not based on ancestry and its biological effects in brain. This is almost more likely environmentally related factors. Next slide. So just a take-home message from the research. I want to emphasize this was a very short survey of this work. It's really quite a comprehensive report that was in Nature Neuroscience 1 month ago in June. We also had the cover art related to that paper, which we were really very pleased with. And there's a lot more in this paper than I could describe. And this is really just phase one of this whole project. We're now digging very deeply into all of these cells that seem to explain these ancestry effects on gene expression and trying to develop algorithms that we can use in clinical studies to predict who is at increased risk of these things. But the bottom line is thousands of genes and transcripts, which are pieces of RNA, are differentially expressed in brain based on proportions of African versus European ancestry in African American individuals. The differentially expressed genes involve immune function and also blood vessels, which I didn't talk about, but not neurons. The majority of ancestry-associated gene expression is driven by DNA sequence variation, which is what ancestry means. There is a relatively minor role for the environment on gene expression, at least once we control for how genetics plays a role in that. And I want to mention that only because we have not explored the possibility, which has to be explored and will be in the next phase of this, that genetics influences how the environment affects gene expression. And then the prevalence of various diseases in African Americans, both their presence and their absence, is based in part on genetic ancestry. We're relentlessly pushing the scientific frontier to discover ways to prevent, treat, and ultimately cure brain disorders. And I think part of our purpose here is to suggest that we are looking for partners, we're looking for participants, we're looking for collaborators to join in this effort. Thank you.
Terrific. Thank you, Dr. Weinberger and Dr. Thomas, for just a terrific presentation. Lots to unpack. I'm going to start with some questions, and then I know we'll be getting questions from our audience. So my first question goes to you, Dr. Hathaway. If we ever needed scientific evidence that we need to include everyone in research, this project certainly has that. And you all had a very sensitive assignment, which you alluded to in the presentation, to convince the relatives of loved ones to donate their loved one's brain matter to science. How did you go about this? I mean, before you made the request, what sorts of activities led up to this? And what can others learn about approaching the African American community and other underrepresented groups in research?
Thank you. One of the real issues that we wrestle with within the African American community is that when President Obama around 2008 began to talk about precision medicine, that medicine could be particularly tailored towards your genomic data set, I recognized then that this was a real social justice issue. I come out of the faith community. I come out of a social justice tradition. So as I began to work with the people at Lieber, as we began to conceptualize this African Ancestry Neuroscience Research Initiative, I said, wow, I have one or two objectives. One, I can take the traditional social justice protest methodology and say, this is not right. Or I could take a different approach, which basically said, why don't we work together, collaborate together to address an inequity that we've identified, which is the dearth of participation of persons of African ancestry in neuroscience research. as I began to work with them, what was very comforting to me, I come from a church, I had a member of my church whose uncle was a victim of the Tuskegee experience. So, so I knew of the pain and the trauma that that family experienced because of that. So I was very sensitive to how were we ensuring consent? How was we ensuring participation? And one of the strengths of the Lieber methodology is that their consent process is very precise, very accurate, and very inclusive. So when I checked that box and realized that the persons who were participating of their own volition, that they were contributing, and then when I looked at the rate of contributions across racial backgrounds, it was basically the same. So persons of African American descent were consenting to the donations. Persons of other ethnic backgrounds were too. So this really assured us as we began to work. But the most important thing that I would say just in my presentation is this. How do you involve the community in this type of research? First, I started with a kind of curiosity. I had a brother who was born with autism. He was on the spectrum. So I always was wondering about, could there something have been done? When I discovered that they had this amazing repository of brain tissue, and then recognized that there was a large repository of persons of African ancestry that were not, the research was not being accelerated upon, that's when I began to get to work, began to advocate within the community. I brought in different stakeholders to get involved. We brought in funding sources to get involved. But most importantly, we had a definition of community that I think was historically valid inside of Baltimore. I was a pastor of Union Baptist Church. Union Baptist Church began in 1852. I went to Morgan State University. Morgan State University began in 1867. So we were talking about fundamentally stakeholders in the building of the African-American community in Baltimore now being involved in this research initiative. And I think that became the key crux, was that the credibility of a faith-based institution, the credibility of historic Black college and university, their connective tissue within the community, we included everyone as we begin to move. So I just, as a way of just saying that, one, I thought it was very important that the consent process was a very documented and accurate and inclusive process. Two, that the community had a real stake and role in the development of the African Ancestry Neuroscience Research Initiative.
Terrific. You know, we often talk about trusted messengers, and I can't think of a more trusted messenger in the Baltimore community than you, Dr. Hathaway. Thank you for taking us through that process. Dr. Jaraza, how can academic and industry leaders better ensure that research subjects are representative of the whole population? You know, what are some of the push and pull mechanisms that could accomplish this?
Yeah, well, first of all, it's an honor and a pleasure to be here with you and amongst friends. Like Dr. Weinberger, I'm a psychiatrist and a neuroscientist. Like the Reverend Hathaway, I have deep connections with Baltimore, going to undergrad at UMBC and family members there. And Dr. Tama, I knew before she was a doctor, actually doing her PhD graduate work in my lab. So it's just always a heartwarming experience to share time with them talking about something that's so incredibly important. Your question around thinking about what academic and industry leaders can do to participate and bring more people in this research space, I'll sort of take a step back and sort of frame it historically. So I never appreciated, when I was going through medical school and graduate school and residency, that there was a time where people were doing clinical trials that only included men, right? The idea of that seems absolutely absurd, that there are clear biological differences that undoubtedly have health relationships. And yet our understanding of health and biology and the creation of medicine was being developed in a framework that didn't include half of our population, right? And if you were to say such a thing now, people would tell you that it was totally absurd, right? That a world like that existed. And ultimately that changed because there were policy changes. Some of them were central at the National Institutes of Health, in which it started to demand if it was going to fund a clinical research trial, it had to include women. And industry also played a huge role in this as well, realizing that there was a marketplace for also figuring out how to create medicines that worked optimally for women. And I remember going through residency, sort of being struck with this. At the time, there were some studies that came out which suggested one of the medications that we were using, helped to use to treat sleep, Ambien, that the right dose for this may have been different in men and in women, right? And so for me, I was suddenly in a framework in which excluding people at these initial stages of developing medicines may actually be doing harm in a medical setting where we're first taught to do no harm. So I give that as an example because I think it really frames why it's so important that our clinical trials include folks of all different backgrounds, right? Certainly, there's no doubt that our genetic and genomic ancestry is linked and anchored into biology through all of the details that Dr. Weinberger presented, right? And so for me, I always start by using that example to say, if we're going to do right and we're going to help everybody optimally, we have to create space for this. Like I described for women, NIH does have policies that sort of advance people towards the goal of making sure that all of our population is represented in the clinical trials that they're funding, right? So I think that that's an important policy change. And I think industry is waking up to the idea that there's a tremendous marketplace and market value to making sure that medicines and the creation of medicines really utilize genetic and genomic information of people of all backgrounds, right? This isn't simply a question of how do we make sure that medicines are fair and help everybody, which I think in and of itself is a reasonable goal. But as Dr. Weinberger mentioned, right, there are resilience pathways that may be different in terms of ancestry. So it may be the case that by studying human variation and the differences across ancestry, you may actually be discovering the treatments that help everybody by enriching our data sets with people of different backgrounds and experiences. There are industry companies that have taken this on. A great example of this is the wonderful Together for Change initiative that was established by Regeneron. AstraZeneca is also involved. Roche is involved in partnership with Meharry, in which they're working to create more neuroscientists, create more clinical researchers, but also gather genetic and genomic information of people of different backgrounds. And so I think that this work is underway. But the advocacy is going to be so critical in people explaining why this is of important value for the NIH and other funding agencies continue to focus on. So again, super grateful to be here.
Thank you so much. So actually, this next question I think is a nice segue. And by the way, I want to just remind our audience to post your questions in the chat or in the Q&A. And I know that we already have some. But back to you, Dr. Hathaway, in terms of how can advocates best promote the importance of inclusivity and diversity in scientific research to policymakers? So have you found, because I know that I believe you received some funding from the state of Maryland.
That's correct.
Have you found specific metrics or individual stories to be particularly effective in making this case? How did you go about that?
Well, first you have to look at the fact that when you talk about advocacy, when you talk about involvement, you first have to make certain that, one, you have real community stakeholders at the table. And I have what I call my 4 R's. My 4 R's deal with this. One, I call it recognition. Recognition that community stakeholders bring value proposition to the process. Second thing is that because of that, they are worthy of respect because of their subject matter expertise. The third thing is important, that you then seek to have a relationship so that the relationship is collaborative and that relationship is quid pro quo, meaning that we all are learning in the process. And then it can't just be for relationship persons, it has to be for results. So that's my 4 R's as it relates to that whole community advocacy process. Inside of this process, we utilize all of that. But most importantly, I believe that you need the data and the scientific information of researchers, but you also need the ability of the community to interpret and communicate what that means to them. And I think we bring both of those places to the table, where I sit around and I'm learning about epigenetics, I'm learning about social determinants of health, But I'm also saying, why is this important? It's important because as our community advances, now let's take a segue. I come from the hometown of Justice Thurgood Marshall. And Justice Thurgood Marshall was asked the question, what is equality? Equality is, he said, that you get the same thing at the same time in the same place. So when you apply that to this area of research, that's why it's important that we have everybody at the table, because that is true inclusivity, that is true equality.
Beautifully said. Dr. Thomas, are there branches of biomedical research that are further ahead in the inclusivity and diversity of its research subjects, or does neuroscience particularly struggle with this challenge?
I would answer that yes and no. I think on the surface level, there are some fields like cancer biology, for example, that would seem to have more diversity in terms of their clinical trials, even the population that talks about their process, their experiences with disease. But even now, as we're talking about the environment, social determinants of health, it's really important that we're not forgetting a lot of that research, a lot of those ongoing clinical trials aren't quite getting to the point of looking at things like what are those stress factors, what are the environmental factors, whether it's pollution, whether it is safe living environments, prenatal health outcomes. There's so many things that we're now realizing are very intertwined in all of health and medicine. And I also think that neuroscience has a very interesting place that we take up in the medical field. And I also love some of the history behind how the branches of psychiatry and neurology came to be. But back in the day, this used to just be one field. And then I believe it was maybe in the 70s or 80s, it split of anything that had the physical symptoms that you could see. So for example, the shakiness or some of those big motor defects became part of the branch of neurology, and psychiatry became more of the field of medicine of the brain, of things that change your personality, but you can't exactly see per se. And I think when we talk about neuroscience and psychiatry, especially in regards to African ancestry communities, there's a lot of stigma from a cultural level that we also have to address as well. And I think that's why scientific advocacy, scientific communication, talking to communities is so important, because there are so many cultures. For example, I'm West Indian. I know Kof is Ghanaian. A lot of people share the same sentiments that it might be very taboo in our households to say something like, so-and-so has depression. And that's a unique hurdle to also tackle and to also make sure that we're addressing with the same level of respect, because people see something like cancer and it's, oh, of course, cancer is serious, but things in the neuroscience research space are equally serious and equally hold the same amount of weight, but require a different level of conversation and intentionality in order to even have the diverse inclusion of subjects and just ongoing research. So I think yes and no, that we do have a lot to catch up to, but then also the fields that seemingly have a lot of diversity also need to do more to address the environmental and social determinants of health.
And
Wow,
maybe I
very, very
can just come in
insightful.
there.
Oh, please, please.
Yeah, I just wanted to give a shout out to the National Institutes of Health and the work being led by Josh Denny and his team over there with all of us. This is some work that came about through advocacy, where they're working to bring Americans in of all different backgrounds, sequence their genomic information, and track their health information to close some of the gaps that we've been talking about, particularly those that Dr. Thomas raised. So efforts are underway.
Thank you. I'm going to turn now to my colleague, Sarah, who has been collecting some really great questions from our audience. Sarah, you want to join us?
Yeah, thanks. And thanks for such a wonderful conversation. My background is actually, my PhD is in genetics. So this has been a really fabulous conversation. And I've learned so much. So it kind of feels like back in the lab today. Yeah, we have so many good questions in the chat. So I will try my best to get as many of them answered with the time that we have. So a couple that I think can kind of be answered together, and we've already started to hit on this topic, is we have someone who's asking advice on how to develop culturally sensitive clinical trials, specifically for Alzheimer's and vascular disease. And then there was another question that asked, what are some of the least appreciated requirements for achieving inclusivity, I think, in clinical research? So we've already started talking about these, but maybe we can comment a little bit further.
Who wants to take that?
Well, Dr. Weinberger, I didn't get to your question, so why don't you take it?
Well, I mean, obviously, I've always operated on the assumption as a scientist that science is one of the ways to cut through a lot of the emotionality related to personal experience. And because science, in principle, is theoretically objective, it doesn't take sides, and it tries to provide information that is beneficial to have. So I think what's missing in a lot of these trials is trust and conviction on the part of the community that this is really in their interests. And I do think that that has to be voiced by people like Dr. Thomas, Dr. Zarasa, Dr. Hathaway. These are the kinds of voices that need to talk to the community about why it's in their interest to care about these things. I just want to say from a scientific point of view, because it wasn't quite mentioned, there's really multiple reasons to level the playing field in research and clinical trials. And it's not just to make it fair, in Thurgood Marshall's words. It's also because by studying different ancestral groups, you learn about illness in all ancestral groups. So what you learn in ancestral groups that are particularly more likely to experience something is maybe something about cause that you couldn't get in a group where that thing isn't as easy to discover and decipher. By the same token, if you study resilience in a group that's more resilient, you may discover clues to novel therapies that will help everybody with that disorder. So the irony of thinking about these disparities in research is not only because they disadvantage the people who are left out, they actually disadvantage the people that are in. And that's another reason that's very important to do this. And I think that's partly why we need to engage all communities. I have to say, Reverend Al can remember this very well. When we first started the African Ancestry Neuroscience Researcher, Reverend Al had several meetings that he'd organized with leaders of the faith-based community in Maryland. And they came to the Lieber Institute. We had a lot of conversations about the reason to do this. I was basically, this white nerd scientist just trying to convince everybody that this was good science and meaningful. It would help public health. And many of the people asked me, how could they feel confident that this was not just an effort to make one group look bad and another group look good? And that's been around forever. And so it's very important that that message has to be spoken to. I always say there's no such thing as a better brain. Every brain is different. Every brain has certain things that make it more resilient and other things that make it more susceptible. Nobody has a unique monopoly on only the things that make you resilient. All brains differ in these things. So, I mean, again, I think it's critical that the communities that are traditionally minority communities that are not included in many of these scientific investigations and trials need to have a great deal of attention paid to their mistrust.
And let me just add by adding something that I think we need to do and something that we need to stop doing, right? So I'll add an example of both. So I became a psychiatrist in neuroscience, largely motivated by understanding mental health because mental illness has deeply impacted my family members. And so the idea of advancing science in a way that doesn't apply to my family is intolerable, right? It is, right? We sort of talked about minimizing emotions in this case. It is emotionally intolerable for me because I want to help my family members. And I think every one of us on here understand how deep of a drive that can be, right? So I think the thing that we do need to think about, and Dr. Hathaway sort of mentioned this as sort of involvement in making sure everybody's at the table, is making sure that like those running clinical trials and running the studies represent the breadth of our country, right? All walks of life, all backgrounds, right? Whether it be someone who's on a farm in Oklahoma or the inner city of Detroit or from Flint or from Baltimore, our clinical trials need to better reflect the leadership and the scientists and the perspectives need to better reflect our country. So that's something we need to do. The thing that we need to stop doing is advancing amazing studies that have groundbreaking results and at the end say, but caution should be applied. This needs to be tested in diverse populations, right? I think we need to stop doing that and do a better job of, in those initial studies, before we generate the excitement and the interest and the hope, we make sure that everybody's included in that first step and not as an afterthought. So I think both of those things are critical.
Well said, as always, Dr. Jaruza. Sarah, I think we've got a few more questions.
Oh,
And
yeah.
we probably have time for 2 more.
Okay, great. So maybe we'll pivot to a little more in-depth science question. And there's a question on what exactly is meant by an ancestry-associated gene, and how do we transfer from that idea of an ancestry-associated gene to identifying causality in terms of health outcome?
So that's a very important question. And again, this work, I think, is comprehensive and very important, but it's not absolutely definitive in every sense of the word. So these genetic associations identify factors that we believe attribute risk to individuals. And so ancestry, we know, is variation in our genome. And variation in our genome contributes to variation in how susceptible we are to illnesses. So if we're studying variation in the genome that affects how genes are turned on and off in the brain, that will be how the brain contracts illnesses, manifests illnesses, or is resistant to them. Genes are, by definition, actually, mechanisms of disease. I always like to say the genes are much easier to study than the environment because they're objective, they're bounded, you can design experiments to test them. The environment's much more complicated, and it's particularly complicated because people's genomes, what we inherit, influence how the environment affects us. Some people are much more resilient to environmental adversity than others, and some of that is genetically driven because the genes are about how our cells respond to things that they experience. So, this business of trying to use genes to predict causes of illness is a step in a direction that can lead to that. There's no question that we understand the cause, for example, of sickle cell anemia. Is a mutation in a gene that makes a certain protein called hemoglobin. We understand how genes that are related to certain ancestries affect the biology of proteins that cause illnesses. Genes are, by definition, the egg. They're not the chicken. They're always the egg. That doesn't mean that genes are the most easily treatable problem. In some ways, they may be very hard to treat. We know that smoking causes lung cancer, but there's a huge genetic component to how much genes influence the toxic effects of smoking. But if you want to have a big public health effect on lung cancer, stop people from smoking. That's much more effective than trying to do anything about their genes. So we have to approach both what we can do with genes to predict people's liability for illness and how we can address environmental components that, as Dr. Thomas said, that contribute to how illnesses are manifest. The social determinants, the environmental determinants of variation in population exposures is a huge factor in variation in population risk prevalence, but so is ancestry. So these are components of the complex, multifactorial reality of common human illnesses. But the genes represent something we can really get our hands around, and we can design drugs based on them, and we can understand mechanisms of disease based on them. And I always used to say that the irony of studying the human genome is it probably gives us the best opportunity to understand the impact of the environment. Because I always like to quote Paul Simon, who said, "One man's ceiling is another man's floor," in one of his songs. And that's the point, that, we all experience the environment differently, even the same environment, because of the biological toolbox that we inherited from our parents called our genome.
Yeah, I'll just add one more piece on there, because I know there are a lot of people in here who aren't geneticists and might not even be scientists. And so this question of how do you know what your ancestry is, might be a little bit complicated to sort of anchor into, right? So, if you sort of think about it in this same framework and you want to know where someone is from, you might ask them to say "you all," right? So if they say "y'all," they're from the South. If they say "yous all," they might be from New Jersey, right? And so the word is the same; it means the same thing, but we can associate where you're from sort of by how the language is pronounced. So your genome is basically made up of 4 letters, right? And there are different ways you can organize those 4 letters and get the same thing out of them. And so what you're essentially doing is you're looking at the genome and you're saying the combination of the letters show up this way in this part of the world over time, right? And so that's all ancestry is. It's basically looking at the combination of the 4 letters and figuring out if you say "you all," "y'all," or "yous all," right? And then basically linking that to a region where people sort of pronounce the word the same way. Doesn't mean the word is better or worse, whether it's "y'all" or "you all." It just means it's a little bit tiny differences.
Thanks.
That's great.
Yeah, Sarah, we've got time for one more.
Oh, yes. I love a good metaphor for explaining science. That's a great one, because I think it is so tricky, right, to make that connection. So yeah, I will sneak in one more. Maybe this would be a best case of the future of this research, but given that there is such genetic diversity of people of African descent, do you see us having to go even further in the populations that we're studying and saying, are we getting significant West African descent in our study populations, et cetera?
Yeah, I think that piggybacking off what Dr. Weinberger said, I really love that he used the example of hemoglobin. So something that we talk about a lot in terms of like red blood cells, a lot of people have heard of sickle cell anemia. And so not only do we know that that is more prevalent in Black people, African Americans, but even when we look at the greater continent of Africa, that is really centralized in West Africa. When we go to Northern Africa, when we start looking at another genetic disorder, this one is called thalassemia, that is also affecting red blood cells, but in a completely different way. And so when we're talking about increasing genetic diversity, increasing subject inclusion, we're looking at countries that really might not be that far apart. We're talking about West Africa versus Northern Africa. So in the grand scheme of the continent, these are closer, but the way that red blood cell, that hemoglobin is interacting is happening in a completely different way. And not only that, but I think when we're looking towards the future of what treatments and treatment interventions look like, we still have to pay respects to the people there and what actually makes sense. In pharmacology, there's a term called pharmacovigilance when you're talking about drug safety and just how this is going to interact with the body overall. I think that there's something that we also need to pay attention to with communities is also paying attention and reverence to aspects of the holistic medication, and even thinking about part of that environmental interactions that might be happening is how do things interact with certain holistic remedies? I don't think that we should be approaching it as we're designing new therapeutics and this is just what the solution is, take it or leave it. We should be approaching things to be integrative with what people are already doing. And so I'm sure you've seen on certain medications, you usually have the instructions on the back and it's like, don't mix this with grapefruit juice. Don't take this with alcohol. We should be considering those same sort of things when we're talking about the natural roots, herbs, plants that people are already taking, the things that are already in people's diets. all of that is contributing to some of those epigenetic changes that we're also talking about, that Dr. Weinberger's mentioned, that Dr. Jaras has mentioned. And then again, those community conversations, the 4 R's that Dr. Hathaway has mentioned. So not just stopping at regional diversity, but taking it a step further of, with respect to regional diversity, what does it mean to approach therapies that make sense for these communities?
Terrific. Thank you, Dr. Thomas, and thank you, Sarah. Before we end, we did get some questions that people sent in, and they really had to do with how can we learn, how can we incorporate your learnings at Henri into my research? And so my question is, we've posted your really great website in the chat, as well as links to some NPR interviews, et cetera. But is there a way for folks, either via your website or otherwise, to connect with you directly? And, you know, are you, you know, do you have a you know, presentation that you've been giving to others in the research community around community engagement? So just if you could share those who are listening how to connect with you all.
I think
really
the best
my question.
I think the best way is through our website.
Yeah.
That's
Okay.
the best, most direct way. The amazing thing is that as you see with my colleagues, they are well versed, but they understand something that I communicate well. We must be culturally sensitive and culturally relevant. And when we talk in those terms, when I'm interacting with my American Indian colleagues, they say that's right. You know, when I'm talking to my brothers in Hispanic orient, they say that's right. So I think our terminology and our language that is inclusive in terms of community also is coupled with the research that we do. So yes, please contact us directly through our website, and I know there'll be a response.
I can't thank you all enough for joining us today. What an inspiring and important project you're leading. Before we go, I just want to let our audience know that if you are a Research America Alliance member, please join us next Wednesday. We're going to be talking about the 21st Century Cures 3.0 with the staffers who are working on that. And we really look forward to your input. Please email my colleague, Jacqueline Lagoy. Her email's in the chat for the registration link if you're an Alliance member. Thank you again for joining us today, both our speakers and our audience. We look forward to seeing you again soon.
Thank you.
Thank you, Nan.